to give you an update on genomic advances in Waldenstrom's and the potential for clinical translation. And so, given the time, I'll just summarize by saying that Jan Waldenstrom, as you know, was credited for this disease back in 1943. And yet, despite the efforts of a lot of individuals, both accepting that this was a bona fide clinical pathological diagnosis as well as one that could be founded in genomics, remained to be seen. And so it was in 2002, in fact, that experts had come together in order to agree that this was a bona fide clinical pathological entity. But it would really be 10 years later that we finally had, a pathological or a molecular basis for this disease. And this was largely based on work involving whole genome sequencing that was performed in our laboratory, used paired sequencing from individuals who had Waldenstrom's against their normal tissue. And so, as many of you know, the key finding from that effort was the discovery of a mutation, a point mutation in the MYD88 gene, one which resulted in a change in leucine for proline at position 265 on the amino acid. This was subsequently confirmed by Sanger sequencing, and what was really astonishing was the fact that over 90% of the patients by whole genome in Sanger sequencing had this mutation. For some patients, about 15% is even a AUPD that exists at this juncture where the mutation is to be found, resulting in a homozygous phenotype. Although most of the people are heterozygous, about 15% can be homozygous for this mutation. And its exact clinical impact remains to be clarified. Now, since the reporting of the whole genome study, many labs across the world have now confirmed the existence of this mutation as a highly prevalent mutation in Waldenstrom's. You can see the prevalence layers. This depends a lot on the kind of technology that's used. Of course, if you use PCR technology, you're more likely to pick it up. And in some centers, up to 100% of the patients who have the clinical pathological diagnosis of Waldenstrom carry the mutation. But what's also interesting is that as we moved from Sanger sequencing into PCR, we also began to see this in most of the patients who had the precursor to Waldenstrom's, IgM monoclonal gammopathy of unknown significance, with upwards to 50 to 80% of patients displaying this mutation. Now, what that tells us is that this is likely to be an early oncogenic driver for the disease, and likely that there are going to be other mutations that end up playing a role. And for this reason, we began to look at other potential contributors. And, to no surprise, we fell back on chromosome 6, an area that Rafi and his group had long studied before us, in fact, documenting that 6q deletions were quite common, in fact, up to half of the patients with Waldenstrom having these. And there were a number of genes that were implicated in this area, including BLIMP1, otherwise known as PRDM1, as well as TNFAIP3. But as it turns out, based on whole genome sequencing, there are other players in this area that probably play even a more central role, with upwards to 90% of patients showing copy number losses, small copy number losses that actually end up affecting these regions. This includes genes like HIVP2, which are very important NF-kappa B regulatory genes. You lose HIVP2, you amplify NF-kappa B signaling. BCLAF1, whose role in BCL2 regulation has been implicated. And even what we found very interesting was even the loss in up to 40% of patients of an inhibitor of BTK. And we're going to talk a little bit about BTK in just a moment. Now, what the discovery of this mutation allowed us to do is to first be able to improve our diagnostics for Waldenstrom. And I think many of you recognize that there are a lot of overlapping disorders that secrete IgM. And in fact, you can see relative to marginal zone lymphomas, even IgM secreting multiple myeloma, certain types of CLLs can also secrete IgM, that there's an absence or paucity of the MYD88 mutation in these entities. Now, more recently, we've tried to see if we could improve diagnostics by moving away from bone marrow testing and focusing on peripheral blood testing. And in fact, this appears to be very much achievable. This is a paper that just came out in Leukemia, where we used paired samples of untreated patients with IgM-MGUS, smoldering, as well as symptomatic Waldenstrom's. You can see, in fact, the strong correlation between peripheral blood testing for B cells, as well as bone marrow testing for B cells for the MYD88 mutation, with very high levels of sensitivity and specificity. In the treated patients, it's a little bit less, and that's because the drugs that we use have a discordant compartmental effect on peripheral blood for many of these patients. What's also interesting is that by looking at the delta CT levels for the mutation itself, it may be possible that we can separate IgM-MGUS from Waldenstrom. And in fact, this was also demonstrated in the leukemia paper, where delta CT of less than 6.5 was seen in the majority of smoldering as well as symptomatic Waldenstroms, but it was rare in the IgM-MGUS. So I think the time has now come that we're going to be able to use MYD88 testing, much like JAK2 is used in PV, that we can even make these diagnoses with simple peripheral blood testing. Now, the other area that has been, of course, of great importance is to see what we could do therapeutically with this knowledge. And this has been the work of Lou Stout's group as well as our group that have largely focused on this, with Guang Yang in our laboratory playing a very important and pivotal role. And what we do know is that MYD88 is an important adapter protein for both toll receptors that sense various pathogen particles as well as interleukin-1. And through stimulation through these receptors, MYD88 homodimerizes and triggers these Iraq molecules to become phosphorylated, which then trigger these downstream signaling, leading ultimately to NF-kappa B activation and translocation of the nucleus. And we know this is central in Waldenstroms because when we use, in fact, a homodimer inhibitor for MYD88, we're able to block translocation of NF-kappa B into the nucleus with the MYD88 homodimer inhibitor. Now, even more importantly, if you use these toolbox compounds that affect either MYD88 signaling or Iraq, which is downstream of MYD88, in Waldenstrom cell lines that carry the mutation, you can induce apoptosis. And you can also see this occurring in primary WM cells in a very selective manner. You see the healthy donor cells are spared. But what intrigued was that we could always get more killing by interfering with MYD88 itself, rather than downstream Iraq signaling. And of course, in the native sense, this is what we know to be true, is that MYD88 signals through Iraq. So for this reason, we began to look at other partner proteins that play an important role in MYD88 signaling. And the one that we identified was actually Bruton's tyrosine kinase. In fact, when you look at these co-IP experiments, it's the activated or phosphorylated form of a BTK that binds to MYD88. And in fact, if you use the drug ibrutinib that extinguishes BTK activation, you abolish this binding with MYD88. And furthermore, if you knock down MYD88, you can abolish phospho-BTK. You can also show very selectively that if you upregulate MYD88 L265P through transfection models, you can hyperphosphorylate BTK, but you don't see that impact with wild type. And further to this, you can also see very selective killing of MYD88 L265P expressing Waldenstrom cells relative to other B cells that express the wild type. So given these findings, we initiated a clinical study with ibrutinib. I'll talk more about this later today, but as many of you know, this has been a very exciting study and led to an FDA breakthrough designation for ibrutinib in Waldenstroms. Now, there are other targets as well, and coming to a meeting like this, you want to excite people about MYD88 pathway signaling. And there are a number of other targets within the MYD88 pathway that we and others now have been focused on for drug development. But it also is important to recognize that many of these inhibitors could be potentially used in combination, even with ibrutinib. This is work that has been done and published by my colleague Guang Yang, showing in fact that Iraq inhibitors show very nice synergy with ibrutinib, both in terms of these synergistic killing models in Waldenstrom cells, and also very nicely showing that you could inhibit NF-kappa B related signaling in a synergistic manner when you combine these two drugs. And this actually gives us at least the thinking that MYD88 signals through two divergent, pathways, both Iraq as well as BTK, and this kind of strategy, looks promising, as does also using PI3 kinase delta inhibitors like CAL101. Separately, as Guang Yang showed at ASH this year, PI3 kinase delta signaling is also stimulated by MYD88 and also shows synergy with ibrutinib. Now, for the next couple of minutes, I just want to focus on the second most common mutation that we discovered in Waldenstroms, and that's these WIM-like C-tail terminal mutations in the gene CXCR4. This is the first time that a somatic mutation has ever been described in cancer. About 30% of Waldenstroms patients are affected, and the mutations actually occur here in the C-terminal domain. And this slide is not meant to be read by you, but to show you how complex mapping out these mutations can be in Waldenstrom's. In fact, now we've discovered over 30 different types of mutations affecting the C-terminal domain. And some of them are nonsense mutations that lead to truncation of the C-terminal. Others are frameshift mutations. But if you affect the C-terminal domain, you actually affect signaling for CXCR4. And this has been very nicely demonstrated both at the clinical level as well as the preclinical level, as I'll show you in just a second. This is another paper that just came out in Blood looking at the impact of these CXCR4 mutations on the clinical presentation of patients. And what actually caught our attention was the fact that those individuals that had the nonsense mutations were the ones to present with higher bone marrow involvement as well as serum IgM levels, particularly the individuals that we see coming in with hyperviscosity crisis. And so in our laboratory, Yang Hao has been focused on trying to interpret what this kind of signaling entails. And the first thing that we can tell you is that when you in fact stimulate these cells with the ligand for CXCR4 using SDF1, what you see is this impaired internalization of the receptor relative to wild type. And even more interestingly, pERK as well as pAKT that play important survival cues in this disease are also upregulated in response to SDF1 in the mutant construct cells, and in fact show prolonged signaling relative to the wild type. Now, why this has been of interest to us is because, as you know, there are drugs that affect CXCR4, and plerixafor, or AMD 3100, is the prototype. And so we began to model this. And the thing that we discovered, which was quite interesting, is that once you stimulate these cells with SDF1, you in fact see in the wild type, abrogation of apoptosis that's induced by ibrutinib. But on the other hand, if you add in, if you look at this in the transfected model with the WIM mutation, in fact, that SDF1, actually ends up once again, reviving these cells, which could be reversed if you look at caspase and PARP cleavage here, which can be reversed when you use a drug like plerixafor. And in last month's Blood, in fact, the first long-term study of using low-dose plerixafor was published by a group at the NIH, showing, in fact, that you can continually give patients with WIM syndrome plerixafor. So this really opens up the prospect of being able to use CXCR4 inhibitors in combination with drugs like ibrutinib. In the closing comments here, I just want to focus a little bit on the individuals that have the MYD88 wild-type status. As we began to model this to try to better understand what these mutations entail from a survival point of view, despite the fact that CXCR4 mutations may entail a more aggressive initial presentation, they really have very little impact, whether you're looking at the wild-type CXCR4 or the WIM-containing patients, very little impact in survival. But it's in fact that minority of patients that are wild-type for MYD88 that appear to be the ones most at risk for death. In fact, looking at the hazard ratio that's adjusted here, that risk of death is tenfold higher. And so what do we know about this wild-type MYD88 group? Well, we know that they actually tend to have high levels of circulating B cells. This is really, in contrast to what you see in Waldenstrom's, where most patients tend to be peripherally B cell lymphopenic. They also show decreased IgHV3 through 23 rearrangements as well as somatic hypermutation. But perhaps what's actually most interesting is that they have mutations in MLL2 that we don't see in the MYD88 L265P cohort. And as many of you know, these are mutations that actually are found in follicular as well as aggressive lymphomas. And it may be because of this acquisition of this phenotype that these patients tend to have more aggressive disease. So in the last comments, we're often, Waldenstrom's is often clumped in with myeloma. I guess this is why we have, Waldenstrom being presented here today. But what I'd like to really tell you is that, when you look at the genome, much like we also look at the clinical phenotype, these are very distinct diseases. And this is actually the summary of the most common mutations found in myeloma patients. And these are the most common mutations found in Waldenstrom's patients. And you see virtually no overlap. And Rafi was very nice to show you a few moments ago at least a circles plot, showing translocations. And I think all of us appreciate in this room very common, very recurring translocations to be found in myeloma, rare and non-recurring in Waldenstrom's. And in fact, this is the summary of all the translocations that we found in 30 patients. You might right away run over here and take a look at chromosome 6 and say, well, treon, I don't know what you're talking about. Look at all those translocations there. This is actually from one patient who had a chromothriptic event. And beyond that, you really see very rare translocations that are to be found in Waldenstrom's and to be recurring. So to summarize here, MYD88 is really perhaps the most startling of all discoveries that came out of whole genome sequencing. It's helping us from a diagnostics point of view. It's giving us, information about the pathogenesis of the disease. It's also present in early MGUS, so we know that this is, likely to be an early oncogenic driver, and that really, as we begin to unravel the genome by doing comparative studies with IgM MGUS versus Waldenstrom, we'll have more answers in terms of what those critical progression factors are likely to entail. The CXCR4 WHIM story is an absolutely exciting one. I think many of you recognize WHIM syndrome as being a syndrome where you see activation as a result of the acquisition of these mutations. This appears to be true in Waldenstrom's and constitutes the first ever description of a CXCR4 somatic mutation in any cancer, despite the fact that CXCR4 is so dysregulated in so many different cancers. The MYD88 wild type status confers poor prognosis and is associated with ML2 mutations, and clearly more work needs to be done for this high-risk population. And then lastly, the reason why this meeting is here is to see what we can do with this information. And I think for Waldenstrom's, there's a clear pathway forward looking at both MYD88 and CXCR4 pathway inhibitors. I just want to acknowledge all the members of our lab who